So I want you to imagine a real life, microscopic Dr. Jekyll and Mr. Hyde.
Oh, that's a perfect way to put it, actually.
Right, because you have these two organisms that are, well, they're practically identical twins. I mean, they share almost all of their DNA. But when they interact with the human body, they cause two completely different, just absolute medical crises.
Yeah, completely opposite ends of the spectrum.
Exactly. Like one is a sexually transmitted infection that quietly destroys reproductive systems, and the other is this sudden, brutal brain and bloodstream infection that can, kill a completely healthy teenager in like 24 hours.
It's terrifying when you really think about it.
It really is. So welcome to the deep dive. Today we are zooming in on a single genus of bacteria, and that is Neisseria. And for you listening, this is basically going to fundamentally challenge how you think about, well, what makes a pathogen a good bug or a bad bug?
Right, because usually we want to put bacteria into these neat little boxes. We want to say, this one is the villain. It causes this specific disease.
Yeah, keeping things simple.
Exactly. But with Neisseria, that entire framework just completely collapses. I mean, this is really a story about how bacteria literally shapeshift to survive our immune system.
Wow.
And, how they manage to outsmart our most advanced antibiotics. And really how the physical real estate they occupy inside your body ultimately determines your fate.
Real estate. I love that. And we are going to trace that story from 19th century medical detective work all the way to the modern day superbug outbreaks that are currently panicking the World Health Organization.
Definitely have a wild ride.
Yeah, so before we can understand how these twin terribles operate today, I really want to start with a visual. If you were to look through a microscope at a smear of these bacteria, what are you actually seeing?
Okay, so you were looking for a very specific classic silhouette.
Oh.
Imagine two tiny kidney beans or coffee beans just pressed together along their flat edges.
Okay, so like coffee beans.
Yeah, exactly. In microbiology terms, they are gram-negative diplococci.
Diplococci meaning like double.
Right. Diplo meaning two, cocci meaning round or berry-shaped. So they naturally occur in pairs, hence the two coffee beans. But the gram-negative part is actually crucial here.
Why is that?
Well, it's a lab classification based on their cell wall structure. So instead of having this thick, heavy armor plating like some bacteria have, gram-negative bacteria have a very, very thin cell wall that's basically sandwiched between two lipid membranes.
Okay, but I mean, why does that matter to someone who isn't a microbiologist? Like, what does that actually do?
Because that outer membrane is what your immune system actually sees. It's what it physically touches. It's also what our antibiotics have to penetrate.
Oh, right. The front line.
Exactly.
Yeah.
And as we'll get to later, that specific gram-negative membrane structure is the absolute key to both how this bacteria kills people and why it is so incredibly difficult to make a vaccine against one of these twins.
Okay, that makes sense. But let's go back to those coffee beans for a second. How did we even figure out what they were? I mean, how did a 19th century doctor look at these identical shapes and somehow connect a sexually transmitted infection with, of all things, infant eye infection?
Yeah, it sounds impossible, right? But it was just pure observational brilliance by this German physician named Albert Neisser. This was back in 1879.
1879, wow.
Yeah, so he was looking at pus smears from adult patients with gonorrhea. And at the exact same time, he was examining smears from newborn babies who had this severe, often blinding eye infection called ophthalmia neonatorum.
Oh, that's awful.
It was devastating. But he noticed that under his microscope, in both the adult genital smears and the infant eye smears, he saw the exact same paired, bean-shaped bacteria clustered inside the patient's white blood cells.
Wait, which means the babies were being infected as they passed through the birth canal?
He deduced exactly that. I mean, he argued that this specific microscopic coffee bean was the root cause of gonorrhea in adults and the cause of this tragic blindness in infants.
But, I mean, that had to be heavily debated back then, right? Because the prevailing standard, Koch's postulates, didn't that mean you had to actually grow the bacteria in a lab dish to prove it caused a disease? He just saw them on a slide.
That is the fascinating part. He couldn't grow them.
Really?
Yeah, Neisseria are notoriously finicky bacteria. They require very specific temperatures, high carbon dioxide levels, super rich nutrients just to grow in a lab. They're incredibly fragile outside the human body.
So Neisser was essentially making a massive scientific leap based purely on just visual correlation.
Exactly. It took another 3 years until 1882 for other scientists to finally figure out how to cultivate it on solid media to actually prove him right. And, the genus was later named Neisseria to honor him.
Okay, so that's the gonorrhea side of the family, right? Neisseria gonorrhea. When does the evil twin show up?
Just a few years later, actually, in 1887, an Austrian pathologist named Anton Weichselbaum was investigating a totally different medical nightmare. There were these recurrent, terrifying epidemics of what they called cerebrospinal fever sweeping through Europe.
Meningitis.
Right. So he took spinal fluid from patients who had died of this meningitis, looked under the microscope, and found a diplococcus.
Wait, but if they look identical, did he think the gonorrhea bug was somehow getting into people's brains?
No, because the clinical context was entirely different. Weichselbaum realized this was a distinct organism causing a completely distinct disease, even though they were visually so similar.
Okay, so they were later classified into the same genus.
Exactly. So now science has Neisseria gonorrhoeae and Neisseria meningitidis. And here is the genomic bombshell that modern science revealed. If you sequence their DNA today, these two bacteria share roughly 80 to 90% of their genetic identity.
That is wild. I mean, we share about that much DNA with a mouse, but for bacteria, I'm guessing that makes them practically identical twins.
Well, it goes way deeper than that. They swap DNA with each other promiscuously.
Like they trade genes.
Yeah, they are so genetically fluid that some evolutionary microbiologists argue we shouldn't even think of them as two separate fixed species. They are more like ecotypes.
Ecotypes.
Yeah, think of them as part of a single, highly flexible genetic pool that just happens to occupy two completely different anatomical niches in the human body. And because of that, they express two very different clinical faces.
Okay, well, let's talk about those niches, because this is the real estate problem we mentioned earlier. If they are this closely related, why does one cause an STI and the other cause a deadly brain infection?
It all comes down to the specific mucosal address they evolved to colonize. And the way they behave at those addresses completely defies our common sense about how sickness works.
All right, let's look at the gonococcus first.
Right, so its address is the genital, rectal, pharyngeal, and conjunctival mucosa. When it sets up shop there, it is an absolute parasite. There is no such thing as a healthy carrier of gonorrhea.
But wait, people constantly say that if you have gonorrhea, you definitely know it. Like, the assumption is that STIs announce themselves pretty loudly.
And that assumption is incredibly dangerous. The gonococcus is very often a silent invader.
Silent, really?
Yes. A massive proportion of infected women, I mean, some epidemiological figures suggest the majority, and a very meaningful minority of infected men experience absolute zero symptoms. No pain, no discharge, nothing.
So people are just walking around feeling perfectly fine, but they are silently spreading it?
Mm.
And beyond transmission, what happens to the person carrying it if they never get treated?
Well, the bacteria doesn't just stay put. It ascends. In women, it climbs into the upper reproductive tract, leading to pelvic inflammatory disease, or PID.
Oh, wow.
Yeah, and this causes profound internal scarring, which leads to ectopic pregnancies and severe irreversible infertility. It causes devastating downstream damage entirely under the radar.
It's basically an invisible wrecking ball.
Okay, now what about the meningococcus?
Right.
Like, real estate is different, right? Very different. The meningococcus lives in the nasopharynx, which is the very back of your throat, behind your nose. But unlike its twin, it isn't always a wrecking ball. Population studies show that somewhere between 5 and 35% of perfectly healthy people are carrying the meningitis bacteria in their throat right now.
Hold on, wait. Up to a third of people just have the bacteria that causes deadly meningitis chilling in their throat, and it's doing nothing.
Basically, yeah.
Why aren't we seeing millions of people dropping dead from brain infections every single day then?
Because invasive meningitis is actually a rare, almost accidental outcome. For those carriers, the bacteria is just peacefully coexisting with their immune system.
So it's just hanging out.
Exactly.
Yeah.
The disease only happens when the bacteria accidentally breaches that mucosal barrier in the throat and spills into the bloodstream or the spinal fluid.
Well, how does it breach the barrier? Do you just get unlucky?
Sometimes, yeah. Sometimes it's a new, unfamiliar strain your body hasn't adapted to. But very often, it's triggered by a preceding viral infection, like getting the flu or a bad cold.
Oh, so the virus weakens the wall.
Right, the virus damages the tissue in your throat, inflames it, and literally gives the Neisseria bacteria a physical opening to slip through the cracks. The scary part is you don't usually catch meningitis from a dying patient in a hospital. You get it from a perfectly healthy person who happens to be a carrier, or honestly, from the bacteria already living harmlessly in your own nose.
That is deeply unsettling. So we have the gonococcus, which is always bad news but hides its symptoms, and the meningococcus, which is usually harmless until it accidentally falls through a crack in the wall.
Well,
How do these identical twins manage to survive in our bodies for so long? Why doesn't the human immune system just recognize them and wipe them out?
because they survive through constant, relentless molecular shapeshifting.
Shade shifting.
Yeah, they are engaged in this intense evolutionary arms race with your immune cells, and they are incredibly good at it.
Okay, I read an analogy for this in the notes that totally blew my mind. It's like a bank robber who walks into a bank wearing like a red jacket and a baseball cap. The security guards, which are your immune cells, they get alerted and they start looking for a guy in a red jacket.
Right.
But while he's still inside the bank, the robber somehow swaps into a blue sweater and sunglasses. So the guards walk right past him, and he just keeps doing this over and over.
That is the perfect way to visualize it. In microbiology, the mechanics of swapping those jackets and hats are called antigenic variation and phase variation.
Okay, break that down for me.
Well, these bacteria have these hair-like appendages on their surface called type IV pili. Think of them as little grappling hooks they use to attach to our cells, and they can actually twitch to pull themselves along.
Twitching grappling hooks, got it.
And they also have these outer membrane proteins called OPA and OPUC, which are like cellular Velcro. The bacteria can literally switch out the molecular building blocks of these grappling hooks and Velcro, physically altering their shape, or, they can just turn the proteins off entirely.
While they are inside you.
Yes. And they do this incredibly fast, like on the time scale of a single ongoing infection.
Okay, I get the what, but I need to understand the how. How does a single bacterium rewrite its own physical outfits that fast? Where is it getting the new hats and sunglasses if it didn't bring them into the bank?
It gets them from the corpses of other robbers.
Wait, what?
Yeah. Neisseria uses a process called natural transformation. It is one of the most naturally transformable bacteria known to science. It acts like a giant genetic sponge.
A genetic sponge.
Right. In your throat or your reproductive tract, bacteria are constantly dying and shedding fragments of their DNA into the environment. The surviving Neisseria bacteria actively suck up this free-floating DNA from their surroundings, and they physically recombine it.
So they weave it into their own DNA.
Exactly. They weave those new genetic constructions into their own genome in real time.
That is terrifying. They are actively crowdsourcing their disguises from the environment.
Yes, they are absolute masters of horizontal gene transfer. And the clinical implication for you as the patient is severe. This is the exact reason why you can get reinfected with gonorrhea over and over again throughout your life.
Because your body doesn't recognize the new disguise.
Bingo. Your body fights it off, your immune system builds antibodies to remember the specific red jacket the bacteria wore last time. But the next time you're exposed, the bacteria is wearing a completely new molecular face. Your immune memory is utterly useless.
That is such a bleak reality. So we have this shape-shifting bug that outsmarts our immune system. How did humanity ever fight back against this before, modern medicine?
We had to get incredibly creative. One of the greatest early triumphs actually goes back to 1881, involving a German obstetrician named Karl Krede.
Okay, what did he do?
He was trying to stop that blinding infant eye infection, ophthalmia neonatorum. He figured out that if he put a single drop of dilute silver nitrate solution into the eyes of every newborn immediately after they were delivered, he could chemically destroy the bacteria before it established an infection.
Wait, he essentially invented a universal preventative protocol for a sexually transmitted infection decades before STIs were even like a recognized category in medicine.
And decades before antibiotics were discovered. It was a massive public health victory. Now, obviously, silver nitrate causes chemical irritation in the eye, so today we've refined it and usually use erythromycin ointment instead.
But the principle is the same.
Exactly. That foundational principle, treating every infant's eyes at birth as a blanket shield against a silent pathogen, remains one of our oldest and most effective medical interventions.
So that was a massive win against the gonococcus. But let's look at its twin. Why is it that meningitis always seems to strike in college dorms? You constantly hear these horror stories about a freshman getting sick and then the whole campus goes into lockdown.
Yeah, that goes straight back to the real estate problem. Meningococcus thrives in the throat. So in dense young populations like college dormitories or military barracks or boarding schools, people are constantly sharing space.
Sharing drinks, coughing on each other.
Right. The carriage rate, which is the percentage of healthy people harboring the bacteria, it spikes dramatically in those environments. And when an invasive strain finally breaches the barrier in one unlucky student, it moves with terrifying speed.
We saw this with the 2013 outbreaks at Princeton University and UC Santa Barbara, right?
Yes, absolutely.
And the sheer severity of it. I mean, the sources mentioned a student athlete at UCSB who survived the infection but required double foot amputations. How does a bacteria in your bloodstream result in losing your feet? I don't really understand the mechanism there.
It comes back to that gram-negative cell wall we talked about at the very beginning. Neisseria has a specific type of molecule in its outer membrane called lipooligosaccharide, or LOS.
LOS.
It's an endotoxin. When the meningococcus gets into your bloodstream and rapidly multiplies, it constantly sheds pieces of this LOS outer membrane.
Oh, like it's leaving a toxic trail of breadcrumbs.
It's more like throwing biological grenade.
Shindig.
Yeah, because the LOS triggers an absolutely catastrophic overreaction from the human immune system. Your immune cells just freak out and trigger this massive inflammatory cascade. This leads to a condition called disseminated intravascular coagulation.
That sounds bad.
It's incredibly bad. Essentially, it's like someone hits the emergency brake on your entire circulatory system. Your blood starts spontaneously clotting everywhere, all at once, inside your microscopic blood vessels.
Wow. And if your blood is clotting inside the vessels, it's not flowing to your extremity.
Exactly. It completely cuts off circulation and oxygen to your hands and feet. The tissue literally dies. That vascular damage is what makes amputations necessary just to save the patient's life.
That is horrific.
It gets worse. It can also cause bleeding into the adrenal glands, which is known as Waterhouse-Friderichsen syndrome, leading to massive hormone collapse and shock. That's why a healthy teenager can die within a day of their first fever.
And this whole time, the bacteria isn't even like actively eating your tissue. It's just shedding a molecule that tricks your own immune system into destroying your blood flow.
Yeah, the human immune system is a major double-edged sword. And actually, speaking of the immune system, there is a fascinating genetic vulnerability that determines who is most at risk for this.
Oh, right. Some people have what microbiologists call an experiment of nature playing out in their DNA. Are you talking about the MSC complex?
I am. So your immune system has a specific pathway called the terminal complement pathway, specifically components C5 through C9. These components assemble into something called the membrane attack complex, or MA.
Okay, the MA.
Think of the MAC as a specialized microscopic hole puncher. Its entire job is to seek out gram-negative bacteria and physically punch lethal holes straight through their thin outer membranes.
So it's our direct counterattack to their specific cell wall structure.
Yes, exactly. But some people are born with inherited genetic mutations where they're simply missing these specific hole-punching components.
Missing them entirely.
Yep. Now, if you have this deficiency, you wouldn't even know it in your daily life. Your immune system handles 99% of normal viruses and bacteria just fine using its other tools. But when it comes to invasive meningococcal disease, you are missing the one specific weapon required to kill it.
So how much higher is their risk?
People with this deficiency face a risk of meningitis that is 1,000 to 10,000 times higher than the general population.
10,000 times higher, just for missing a single microscopic hole puncher.
Yes. It is one of the clearest examples in all of medicine, linking a very narrow genetic defect to a vulnerability against one specific genus of bacteria. It's so well documented that today, if a doctor needs to prescribe you a complement-inhibiting drug for, say, a rare blood disorder, a drug that intentionally turns off your MA complex, they are legally required to give you a meningococcal vaccine first.
Because the drug artificially creates that exact same 10,000-fold vulnerability. Wait, that brings up the biggest puzzle here. You just mentioned a meningococcal vaccine. We successfully used vaccines to fight those dorm outbreaks at Princeton. We have conjugate vaccines that literally wiped out meningitis epidemics across the entire meningitis belt in sub-Saharan Africa.
We do.
If these two bacteria are 90% identical, why in the world is there no vaccine for gonorrhea? We've been trying for a century.
It all comes down to a structural shield. Meningococcus, the brain infection bug, has a thick, stable capsule made of complex sugars surrounding it. That capsule is how we categorize it into groups like A, B, C, W, and Y.
Okay, a sugar capsule.
Right. But more importantly, that capsule gives our vaccines a stable, unmoving target. The vaccine trains your immune system to recognize the sugar capsule, so when the real bacteria shows up, your body destroys it before it can cause harm.
So let me guess, the gonococcus doesn't have a capsule.
The gonococcus is entirely naked. It completely lacks that stable sugar capsule. So when vaccine developers try to target it, they have to aim at the surface proteins underneath. And when they aim at the surface proteins, they run face first into the bank robber problem.
The shape-shifting.
Exactly. The rapid shape-shifting of the pili and the OPA proteins defeats every single vaccine attempt.
The target is always changing faster than the vaccine can train the immune system.
So we have no vaccine for gonorrhea, which means for the last 100 years we have had to rely entirely on antibiotics. But based on the alerts coming from the CDC and the WHO, that strategy is completely collapsing, isn't it?
It is a terrifying sequential collapse. Neisseria gonorrhoeae has systematically defeated every single drug we have thrown at it. In the 1930s and 40s, sulfonamide drugs worked beautifully until the bacteria absorbed resistance genes and made them useless.
Using that genetic sponge trick. Exactly. Then penicillin was the miracle cure for decades, until strains that produced penicillin-destroying enzymes spread across the globe. Then we moved to tetracyclines, and they failed. Then fluoroquinolones, and they failed in the exact same way.
I remember seeing headlines a few years ago about a man in the UK who contracted an extensively drug-resistant case of super gonorrhea. If all those drugs failed, what is our last line of defense right now?
We are essentially backed into a corner. The CDC has designated drug-resistant gonorrhea as an urgent public health threat. It's in the same category of global alarm as multi-drug-resistant tuberculosis. Today, the primary treatment is a single, painful, high-dose injection of an advanced antibiotic called ceftriaxone.
Just one drug left. That feels incredibly fragile.
It is. In fact, for a while, doctors used a two-drug combination, which was ceftriaxone plus a pill called azithromycin. But a few years ago, the CDC officially dropped azithromycin from their primary recommendation.
Wait, did they drop it because it stopped working?
No, they dropped it purely to hide it from the bacteria.
Seriously?
Yeah. They realized that if we keep exposing the gonococcus to azithromycin, the bacteria will eventually use its genetic sponge trick to learn how to defeat it. By dropping it from the standard treatment, we are desperately trying to steward our last remaining drugs and slow down the inevitable resistance.
We're literally hiding our weapons because the enemy learns too fast. That's intense. But amidst all this doom and gloom, there is actually a genuinely surprising twist in this story, right? A silver lining that came completely by accident out of New Zealand.
Yes. This is where the sheer weirdness of genomics offers us a lifeline. Years ago, New Zealand rolled out a massive public health campaign using a specific meningococcal B vaccine called Bacterial to stop a local meningitis outbreak.
Okay, Bacterial.
Right, and this vaccine was unique because it didn't just target the capsule. It also targeted proteins on the outer membrane of the bacteria. And epidemiologists monitoring the population noticed something they could not explain.
The people who got the meningitis vaccine suddenly stopped getting gonorrhea.
Yes, diagnoses of gonorrhea dropped by 30 to 40% in the vaccinated cohort.
That's amazing.
It really is, because the vaccine was never designed for gonorrhea. But because of their evolutionary kinship, that 80 to 90% shared DNA, the two twin bacteria happen to share certain outer membrane proteins. The meningitis vaccine provided an accidental backdoor cross protection against gonorrhea.
A total accident of genetics gave us the first real defensive shield against a superbug.
And it has completely reignited the field of gonorrhea vaccine research. We now have clinical trials exploring how to intentionally use meningococcal vaccine platforms to attack gonorrhea where it's vulnerable.
That is just incredible. So, to bring this all together for everyone listening, we've explored the twin terrors of the Neisseria genus. On one hand, you have the gonococcus, right? A naked, shape-shifting, silent spreader that has defeated every vaccine attempt for a century, crowdsources its disguises, and is actively pushing us to the brink of untreatable antibiotic resistance.
True villain.
Yeah. And then on the other hand, you have the meningococcus, which is this accidental assassin that lives peacefully in the throats of up to a third of healthy people, but can kill within a single day if it slips through our defenses and triggers a catastrophic immune overreaction.
It is a profound reminder that our relationship with the microbial world isn't static. It's a constantly shifting biological war. I mean, the exact same mechanism that allows a bacteria to quietly share our airway can be the weapon that causes a devastating systemic collapse.
It's wild. And as we wrap up, I want to leave you with one final mind-bending fact that completely shatters this good versus evil binary we've been talking about. We've spent this entire deep dive focusing on the dangerous twins, the Jekyll and Hyde. But there is a third cousin in this bacterial family called Neisseria lactamica.
Yes.
It lives in the throats of young children, and it is entirely harmless. It never breaches the barrier and never causes meningitis. But scientists are now actively studying it, and what they're finding is revolutionary.
Because it's acting as a natural vaccine.
Yes. Because it's in the same family, it shares many of the same surface features as the deadly meningococcus. When a child's immune system encounters the harmless lactamica in their throat, it essentially runs a low-stakes training drill.
The immune system learns to recognize the family traits without the child ever actually getting sick.
Exactly. So later in life, if that child is exposed to the deadly meningitis strain, their immune system is already trained and primed to fight it off.
It turns our entire understanding of infection upside down.
It really does. I mean, what if belonging to a pathogenic genus isn't about being a villain at all? What if these bacteria are just playing different roles in a massive, complex microbial ecosystem that is constantly trying to balance itself? The harmless cousin trains the host to survive the deadly cousin. The deeper we look, the more we realize that our simple categories of good bugs and bad bugs don't even begin to capture the brilliant, terrifying complexity of the microscopic world. Thank you for joining us on this deep dive, and keep questioning the unseen world around you.